Go. Okay, we're live. All right. Hey, Bio 375 class. We're going to go ahead and jump right into it with today's lesson 3. And we're going to start with question 1, obviously. So, question 1, this question concerns epigenetic inheritance. So for A, this has to do with genomic imprinting in a human disease called Angelman syndrome. And in the case of Angelman syndrome, the imprint silences the paternally inherited alleles. Suppose little a is the Angelman syndrome allele, while capital A is the non-disease allele. And so the following cross is done, a heterozygous male and a heterozygous female, and it asks for the genotypes and phenotypes of the progeny. And so in this case, because the imprint silences the paternal inherited alleles or the father's alleles, the mother's alleles are the only ones that will be expressed in the phenotype. And so as far as the phenotype goes, half of them will have Angelman syndrome because the mother is heterozygous, and then the other half will be non-diseased, receiving the other allele from their mother. And then the genotypes are, as you can see there, a quarter of them will be heterozygous dominant, half of them will be homozygous dominant, half of them will be heterozygous, and a quarter will be homozygous recessive. But the phenotype is different because of the genomic imprinting. For question B, this concerns a situation presented in part A. So the following cross is done, a heterozygous male with a homozygous recessive female, and it's asked for the genotypes and phenotypes of the progeny. And so because the father doesn't really play a role in the phenotype of the progeny, 100% of the offspring will have Angelman syndrome because the mother is heterozygous for the recessive allele. And so the genotype is as follows. Half will be heterozygous and half will be homozygous recessive, but 100% will have Angelman syndrome. And then in C, this is talking about Barr bodies that are created from X inactivation. And it talks about a woman that has triplo X syndrome. So she has 3 X chromosomes. However, the XX, I think that's the way to say it, I'm not 100% sure, have been deleted from 2 of the chromosomes in each of her cells. So how many Barr bodies are found in each of these women's cells? The answer is there would be 0 Barr bodies found, because in order for a Barr body to be formed, there needs to be more than one XX on the X chromosome, because it's the XX that are counted, not the X chromosomes. And so because there aren't any, because there's only one XX, that's not going to delete it because it's only counting one X chromosome. So it's not going to inactivate that one. And then for D, it says, suppose you isolate a cell from a female cat with 2 X chromosomes. However, the T6 genes on each X chromosome are non-functional, while the XIST genes are on each X chromosome are functional. And how many Barr bodies are expected to be found in the cell? The answer to this is there will be 2, because active T6 determines if XIST inactivates it or not. So if it is non-functional, then it cannot stop XIST from inactivating both. So essentially the T6 prevents the XIST from inactivating the other X chromosome. But in this one, because the T6 is non-functional, it doesn't prevent the inactivation from happening. So there's 2 Barr bodies that are created. So that's question 1. I'm going to switch it off to Haley for number 2.
All right. So number 2, basically all the answers were found in the notes of the PowerPoint. So let's go A. So SRY, I don't know if it's SRY or SRY, is an example of a holoandric gene. And Mike2, which is the pseudo-autosomal, is an example of a gene found on both chromosomes, the X and the Y. And for C, a cross between a heterozygous male and a heterozygous female is going to yield 50% normal progeny and 50% dwarf, because it just goes off of the male's genotype. So let's see. So D, in the sex-influenced pattern of inheritance, an allele can be dominant in one sex while the same allele is recessive in the other. So let's go on to E. It's genomic imprinting is an epigenetic process that occurs during gamete formation, while dosage compensation is an epigenetic process that occurs during embryogenesis. And that is found in the PowerPoint as well. And let's see. F, the answer is X-ist and T6, which is found on slide 16 of the PowerPoint. And for the last one, G, when reciprocal crosses give identical results, the first blank is going to be autosomal chromosomes, and the second blank is going to be the X chromosome. And that's because if it is X-linked, it's going to be, the progeny is going to be different when you do the reciprocal crosses. And that leads us to number 3.
Perfect. Okay. So for number 3, it says that a woman's father has ornithine transcarbamylase deficiency, OTD, an X-linked recessive disorder producing mental deterioration if it's not properly treated. So the woman's mother is homozygous and is unaffected. So what is the genotype of the woman's father? What is the genotype of the woman's mother? So the woman's mother, for question A, the genotype of the woman's father is X. I'll show you right here. So I chose the alleles lowercase d and uppercase d, lowercase d being the recessive allele for OTD. And so the woman's father is affected by OTD, and he has the genotype of an X-linked recessive allele and a Y, obviously, and then the mother has 2 dominant alleles for normal and 2 X's for that. If the woman has children with a man who does not have OTD, what is the chance that their first child will be a son with OTD? So in this case, oh, I wrote this down wrong. Dang it. Hey, Haley, did you do B and C? Because I did them incorrect on here.
Yeah, I got you. So
Okay,
let's see. So if the woman has children with a man who does not have OTD, what is the chance that their first child will be a son? So we know that 2 out of the 4 of their kids will be boys. So we factor that in, and we know that 2 out of 4 of all the boys are going to be affected, because the cross gives us a genotype of X with a big T and X with a little t for the boys. And so half of them are going to be affected, which gives us that the chance that their first child will be a son with OTD is going to be 4/16 or 1/4. And let's see. C2, right? All right.
Yeah, that'd be great.
All right. So the genotype of a male with whom the woman could produce a daughter with OTD would have to be an affected male that has a small t on his X chromosome, because that's the only way that the daughter can have 2 small t's on both of her X's.
Okay, and then I did D, so sorry. Okay. Those other 2, I misunderstood the question.
You are just fine.
Okay, and then for D, so consider the cross described in part C. So because it needs to be with the male who is affected, the expected genotypes and phenotypes of the offspring. So actually the genotypes and phenotypes have the same probability. So a quarter of them will be affected males, and an affected male's genotype is X with the recessive allele and then Y. And then a quarter of them will be unaffected females, which is would be a heterozygous, both X's because they're female, and then a quarter will be unaffected males with a dominant D-linked or dominant allele linked to the X, and then a quarter of them will be affected females. And that's obviously both recessive alleles on the X genes. So you can see that kind of right there. I wrote that out. Okay, and then question 4 for Haley.
All right. So the presence of a beard in some species of goats is determined by an autosomal gene that is dominant in males and recessive in females. So this question's kind of fun. So for A, I saw this. I don't know how well you can see that. So A and B have the same crosses. So it's the same cross, and you just get different progeny out of it, which is kind of nice. So you have 2 heterozygous parents, and that yields, I've outlined the bearded males in blue and the bearded females in red. So because it's recessive in females, they're only going to have beards if it's 2 small d's, which means that this cross gives us not only a 3-to-1 beard to no beard ratio in males, but also a 1-to-3 beard to no beard in females, which is nice. So the answer to A and B is both parents are heterozygous. And for C, I did the same thing here where the males with beards are outlined in blue and the females are outlined in red. So we have the mother would have to be heterozygous while the father is homozygous recessive. For D as well, because it says that both lack beards, that the father is heterozygous and the mother is heterozygous and the father is homozygous recessive. And that gives us this as well, which gives us a 1-to-1 ratio of bearded to not bearded. And on to number 5.
Perfect. Okay. Number 5. This involves X-linked, X-linked inheritance and reciprocal crosses, and we'll go through this pretty quick. So in cats, an orange color is determined by the recessive B allele, and black color is determined by the capital B allele. The heterozygous condition produces a phenotype known as tortoiseshell. The B and the capital and lowercase b alleles are X-linked. Suppose you cross an orange cat to a tortoiseshell female cat. What are the genotypes and phenotypes of the progeny? And so if you look here, this is the father and mother that are crossed, and a quarter of them will be male black because it's an X-linked thing. A quarter of them will be male orange because they have the lowercase b, and a quarter of the females will be orange, 2 of the recessive alleles, and a quarter of the female will be tortoiseshells because heterozygous produces an incomplete dominance effect, I believe is what it is, or codominance. I'm not sure what tortoiseshell is, but yeah. So, and then B, it asks, male tortoiseshell cats are very rare. What is the genotype of a male tortoiseshell cat? And so for this, I believe it can only be in a male with Klinefelter syndrome. And Klinefelter syndrome is when a male has 2 X's, because he needs to be heterozygous, but because it's X-linked, he can't, like a normal male can't have that because he has an X and Y. But for Klinefelter syndrome, you have a male will have 2 X's, and so it would be possible in that case, but it's very rare. And then C, In fruit flies, a mutation produces the scallop phenotype. Assume that the scallop phenotype is a result of an X-linked recessive mutation. So indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done. So reciprocal cross is when you will cross a homozygous dominant female with an affected male, and then you'll cross an affected female, which is homozygous recessive, with a non-affected male. And when you do that, you get, for this cross, you'll get 100% normal, with half being heterozygous females and half being unaffected males. And then with this cross, you will get 100% normal females and 100% scalloped males. Half of the females will be heterozygous but normal, and then half of the males will be affected. And then the last one, again, the scalloped phenotype. So assume the scalloped phenotype is a result of an autosomal recessive mutation. Indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done. And so for this one, this is kind of the purpose of reciprocal crosses, to determine autosomal or X-linked inheritance. And so this one isn't X-linked. So you do the same like principle as up here, but this time they're not X-linked, and so both the male and the female have both alleles. And when you do the first one, 100% are heterozygous and 100% normal males and females. When you do this one, you have 100% normal males and females as well, so it's exactly the same. So this is when you know it's autosomal. And yeah, that concludes our lesson for today. Anything else, Haley? All right, sweet. See you guys later.